book is getting thinner and thinner. Ooh, bing. It's already April 14th. And 1 month from today, you'll be on vacation. Well, unless you're taking a summer course, right? So, wow. So, let's hit it. Let's get this done. Urinary system. What's the urinary system for? From where? It filters your blood, gets rid of waste. Remember, blood is going to transport not only nutrients and respiratory gases, like we've already talked about this semester, but it's also going to help transport waste. And it's a closed system, so it's just going to keep circling around and circling around. Somebody's going to help remove those wastes from the circulatory system. And the renal system is going to do that. As far as the anatomy of the renal system goes, it's probably one of the easiest ones to study. Because what do we have? We've got a couple of filtering units, kidneys. Actually, inside the kidneys, we're going to find the filtering units called nephrons. And then, what are we filtering out? What do we call that stuff? Waste, but what do we call it? Urine. So we need some place to collect that urine. So we have a couple of tubes that are going to allow the urine to flow from where they're filtered, the kidneys, down into a holding tank. And that's your what? Bladder. The ureters are the tubes that are going to deliver urine that's formed in the filtering unit down from the unit to the holding tank. And then we have a way out. Urethra. Good. There's kidney anatomy. Well, there's a renal system anatomy. Let's look a little bit closer at the kidneys themselves. Now, remember, what kind of organs are these? They are retroperitoneal organs, which means what? So, here's the diaphragm, and below the diaphragm we have membranes. Yes? What are those membranes called? They're serous membranes, peritoneal membranes that enclose all the what? All that stuff down there, all the peritoneal organs. And behind that group of membranes, we're going to find the kidneys. So they are here. Are they anterior or posterior? Posterior. We find them here in the back. They don't have a huge, where's my little pointer? Oh, there you are. They don't have a huge amount of protection. A little bit from the last of the ribs, and those ribs aren't even true ribs. What do they call them? They call them floating ribs. They're also called false ribs. They don't have a huge amount of protection. So one of the things I'm going to find in that area, the protective area of the kidney, is a lot of cushion. I'm blind today, so I can't see my pen. What's that stuff? Fat. And that's important. So fat is going to be not only providing energy like it does in all the other places that we've talked about it, but it's also going to be very important to provide protection for those retroperitoneal organs. And one of the reasons they need so much protection is they are highly vascular. And if you look at just these main blood vessels that run into them, we see the inferior vena cava and part of the aorta running down and feeding major branches into the kidneys. So there's quite a bit of blood volume that's running to the kidneys at any given time for filtering as well. So, what does the internal anatomy of your kidney look like? Well, these guys are tons and tons and tons and tons of little tiny filtering units. And the filtering units are going to be in these regions here. They're microscopic, so we'll have a look at them in a minute. But as they filter blood and form urine in these regions called the renal pyramids, kind of in the middle region, and what do we call the middle region of anything? We've learned this term before when we talked about the brain, medulla. So these pyramids, these little regions here, in the middle region of the kidney, the renal medulla, is where filtration is going to take place. Now when I get all of the filtrate, or what's another name for that again? Urine. Where's it going to go? Well, it's going to pour down these little tubes that come off of the pyramids. They're called the calyxes. And they're going to dump into this little holding region here called the renal pelvis. The renal pelvis is formed of major calyxes that kind of connect the minor calyxes from the pyramid regions. And then what's going to happen? So this area here, this middle region where the major calyxes all kind of combine, is called the renal pelvis. Then what's going to happen? I'll give you a hint. They're up here. Your holding tank down here. Yeah. So how's that going to happen? Up here, down there. Yeah. Gravity's going to kind of have everything flow down the ureters to the what? The urinary bladder, the holding tank. So we see different portions then of the internal anatomy of the kidney. You should know where the renal cortex is. Where's the renal cortex? Cortex, just like the brain, that's on the outside. Then we have the medulla, the calyxes, minor and major, to form the renal pelvis, and then down the ureter. Yes? Blood and nerve supply. There's a lot of them. Very highly vascular because I have to do what? I have to filter my whole blood volume, and I do that many times during the day to get rid of waste products. So this is just a feel for how very highly vascular each of your kidneys is. You have two, by the way. Just like you have two sets of lungs, you have two kidneys. How many do you really need? One. Can you live with just one? Yeah. Anybody in here have just one? Or know somebody with just one?
My sister has one.
Yeah. So you can get away with one. Two's nice because you can filter more blood more efficiently. And again, they're very highly vascular. So we get a feel for how vascular they are with this diagram and some of the major vessels that feed in, and how very highly vascular this region is. Now, why are there so many tiny blood vessels there, do you think? Tiny blood vessels. Tiny blood vessels, capillaries. What's happening there? This is where exchange takes place, right? When we discussed the cardiovascular system, we talked about the capillary beds and the fact that that's where exchange takes place. Well, that's where exchange takes place in the kidneys. So we see many, many capillaries in the renal cortex and feeding into this portion here, the renal medulla and the pyramids. Well, guess what's there? Nephrons. And what's a nephron? Well, a nephron is a series of tubes where the whole filtration process is going to take place. Now, the filtration process of blood involves a couple of different steps. Not just taking stuff out, but we actually put stuff back too. And here's kind of the gist of how the nephron works. Up at the top, we see a blood supply. So we see some of those major arteries and veins. And this, pay attention to this diagram because we see this is the cortex, the renal cortex on the edge. So part of the nephron is in the cortex. And then part of the nephron, kind of dips down here, is in the medulla. So when we find these structures, they're kind of hanging out between the pyramids in the renal cortex and the renal medulla, kind of half and half. We see blood coming in to an area called the glomerulus. And what happens there is most of the fluid gets sucked out into these tubes. Actually, way too much gets sucked out into those tubes. And then what's going to happen, as that filtrate passes through this many winding tubes, and guess what we call them? If I have lots and lots of bends and winds, I'm convoluted. There's some convoluted tubules near the glomerulus. Guess what they call them? They call them proximal convoluted tubules. Then we have this little dip down in the nephron called the loop of Henle. And then we have more twisty-turny tubes further away from the glomerulus called the distal convoluted tubules. And it's kind of like a still, except this still is going to actually put stuff back into the blood because it's sucked too much out in the glomerulus. And by the time we're finished, the end product is going to go into an area called the collecting duct. These collecting ducts are going to feed into the minor calyxes, which then dump into the major calyxes, which form the renal pelvis, which then dump into the ureters. And what's left here is going to be what? Nope. Sent to the bladder. This is urine. So we're going to fine-tune that filtrate through these tubes. A better look at what they actually look like is this. I'll go back to the other one in a moment. But they're very highly vascular. So we see a very extensive blood supply surrounding all those tubules. And what do you think is going to happen there? In this diagram, we're showing you all the different kinds of cells that line these different portions of the nephron. And this is where the whole filtration process and fine-tuning that filtrate is going to take place. So, the glomerulus, or Bowman's capsule, is at the beginning of the nephron. Then we run into the proximal convoluted tubules. We dip down into the loop of Henle. Then we have the distal convoluted tubules. And then whatever's left that hasn't gotten put back into the circulatory system ends up in the collecting ducts as urine. So we're going to talk about all of these guys and what happens in all of these areas. Now, these are some scanning electron micrographs of some of the cells that we're going to find in renal cortical tissue. But there's two different types of nephrons that we're going to find in your kidneys. One of them hangs out mostly in the cortex. Remember, the cortex is above those two blood vessels that are running horizontally. Yes? And then we have one type of nephron that has most of its apparatus that hangs way down into the medulla region. The little one over there is called a cortical nephron. And this one over here, you know what it's called? It's got a big long name. It's called the juxtamedullary nephron. And as we talk about what happens in the different parts of these nephrons, we'll understand why we need both types of nephrons, mainly for water balance. Because we're going to do a lot of that. Water and ion balance, that's also a very big job of the kidneys. A lot of that's going to happen down in the loop of Henle. So who do you think is going to allow me to fool with water balance and ion balance a little bit more? Which of those nephrons? Yeah, the juxtamedullary, because of the longer loop of Henle. So, nephron capillary beds is what the textbook talks about next. So you need to know the difference between a cortical nephron and a juxtamedullary nephron. Now what happens is we have tiny, tiny little blood vessels, little capillary beds that form a ball that are going to feed into the glomerular region. These are called glomerular capillaries because they feed into that little cup, the glomerular capsule. Another name for it is Bowman's capsule. And we have arterioles that feed in to this capsule, and this is kind of the place that sucks everything out. This is that little filtering, beginning filtering unit. This is the filtering of the blood itself. So we have an arteriole that feeds in to this ball of capillaries called the afferent arteriole. And by the time things leave the afferent arteriole out the what? Efferent arteriole, things are going to look a lot different as far as blood goes. What am I going to be missing a lot of by the time blood leaves this ball and goes out the efferent arteriole? A lot of fluid. Now, if I was just to take that blood now and throw it back into the general population of blood, would that be a good thing? No, because I'd have way too little what? Yeah, fluid. My plasma would be highly depleted. So that efferent arteriole is the efferent arteriole that is then going to form that loop of little arteries or capillaries that are going to run around the tubules that we just mentioned. Okay? Everybody with me? One of the things we're going to find here are some very important cells that are going to help with that sucking out process. We see groups of cells called mesenchymal cells. And we also see these yellow ones here that are going to help with the filtering process called podocytes. And podocytes create this wave-like motion. Now, if I took a bucket of water and I created a wave-like motion in this direction, where would the water go? Over there, yes? So those podocytes are going to help suck out that fluid from the blood that is running through the glomerular capillaries. There's little feet-like projections. In this diagram, we see them called cytoplasmic extensions that wave and help suck out, create enough pressure to suck out a lot of fluid in the glomerular capsules. Another thing about these capillaries is they're what? They're fenestrated. They have a lot of what? Holes or pores or windows. Remember we talked about that when we talked about the circulatory system? So this is where we're going to find that type of capillary. Why do you think that's important? So I can suck out as much fluid as possible. Do I want to suck cells out of the circulatory system here? No. I just want to filter that blood plasma. Leave the cells alone. They have a job to do inside the circulatory system. So the glomerular capillaries in the Bowman's or glomerular capsule, surrounded by these podocytes, is where a lot of the beginning of that blood filtering is going to happen. And again, we're going to suck out lots and lots of fluid, and then that fluid is going to travel through the efferent arteriole around the rest of the nephron. So they talk about the juxtaglomerular apparatus and the filtration membrane on page 968 and 969. We have a group of cells called the macula densa. And I think we saw that in this diagram, right in this region here. And what are those doing for us? Anybody know? Yeah, they're chemoreceptors. And they respond to sodium and chloride concentrations. What does that have to do with filtering water? Anybody know? Sodium and chloride are charged particles. Water is a polar molecule. Anybody? If I move sodium and chloride or charged particles in a certain direction, water follows. What's that called? We talked about, I know we talked about it. Well, maybe we didn't talk about it. Obligatory water reabsorption. And that's a lot of the principle of getting water balance correct in the nephrons, in the kidneys. So the macula densa is a group of chemoreceptors that are going to help control the process of filtration in the nephron down the line, especially in that loop of Henle where our water balance is going to be dealt with, and also ion balance, and importantly, hydrogen ion balance. Why is it important to balance my hydrogen ion concentration? Yeah, blood pH. So not only are the kidneys responsible for filtering out wastes, kidneys make hormones. What hormone? They're affected by all of them. Remember erythropoietin? Yeah, so they make hormones. They have to filter out waste. They have to take care of water balance. They're also responsible for helping to maintain blood pH. So they're very, very busy, those nephrons in the kidneys. So these are a picture of those podocytes, and the filtration slits between those little feet is where we create that suction, and the podocytes can help with the filtrate process. So what we're going to see is a three parts, of the filtration membrane itself. So we have the capillary, we have the filtration membrane, which is the capillary endothelium. And what's endothelium? Excuse me. Silence. The who? That simple squamous epithelial cells that line the insides of the capillary beds. The basement membrane, which connects all epithelial cells to the next tissue underneath. And those podocyte feet of the glomerular capsule. And those are the apparatus in which we suck out or filter much of the fluid from blood into the glomerular capsule space. So that's the filtration membrane that they talk about. Excuse me. So basically what's happening in the kidneys, it's a three-part process. First, we suck everything out. Too much, as a matter of fact. Not everything, but most of the blood plasma, the liquid, gets sucked out into the glomerular capsule. Then, in the efferent arterioles that run around the tubes, and guess what we call those guys? Yes, we call them peritubular capillaries around the what? Tubes, capillaries. We're going to have a process where we take back into the circulatory system the necessary components. That's called tubular reabsorption. And then, before everybody leaves the whole unit, we're going to fine-tune the process. So we might actually put some things back into the tubules. Maybe we sucked out a little bit too many hydrogen ions, or maybe a little bit too much water back into the circulatory system. We're going to fine-tune the mix down that loop of Henle and in the distal convoluted tubules. And then everybody's going to go back to normal into the major circulatory system again. And the things we put back is a process of tubular secretion. So the whole thing is glomerular filtration, tubular reabsorption, and tubular secretion. Yes? And what we're going to end up with is blood back into the circulatory system that's been filtered. And we're going to end up with a filtrate we call what? Urine. Everybody with me? So glomerular filtration is the first step they talk about, which starts on page 970. And that is due to changes in, and here we go with the whole chemistry thing again, pressure set up by water. Where do you want to go? From a high pressure to a low pressure? You want to go from a high pressure to a low pressure. So what gets set up in the glomerular capillaries is a high pressure gradient. So that the pressure inside those glomerular capillaries, because of the way that they are arranged and set up in the capsule, is much higher than what's outside those capillaries in the glomerular capsule itself, or Bowman's capsule. So due to hydrostatic pressure and the fact that I have those podocytes and the fact that I have these big windows, what's going to happen to fluid from those glomerular capillaries? And that's just one part, hydrostatic pressure. What else? What else is going to be different? Blood colloid osmotic pressure. It's going to be higher inside what? Than in here. Yep. And what's going to happen? We're going to pull things out. And capsular hydrostatic pressure is also going to be different in the capsule than it is in the glomerular capillaries. So what's going to happen to fluid in the glomerular capillaries and glomerular capsule? because of all these differences in pressure. They're going to get sucked out into the glomerular capsule. So if you look at the net filtration pressure, and I'm not going to make you memorize these numbers because I just want you to understand the principle. We can get a little confused, but the net push out of the capillaries is greater than the push in, so we're going to end up with getting a lot more fluid out of the capillary beds than gets pushed in. I know, because you're looking at those going, well, there's arrows going in both directions, but what do you think's greatest? Okay, so the net glomerular pressure, the net filtration pressure is much greater. And we're going to end up with pulling out a lot more than we try to push back in the glomerular capsule. Does that make sense? So these are, this diagram is entitled The Forces Determining Glomerular Filtration and Filtration Pressure. So the net out is going to be a lot greater than the push in. So we're going to suck out a lot of fluid in glomerular filtration. So what's going to help regulate glomerular filtration rate? We already talked about pressure, right? Pressure helps to regulate a lot of stuff in capillary beds. So if you look at page 972, we see some of the different regulations of glomerular filtration described for you. So autoregulation, renal autoregulation, is going to be controlled by many of the hormones you've met already, along with pressure. So the first one they talk about is something called a myogenic mechanism. And what's that? What's myo? Muscle. So muscles around some of these tubes pushing some of this stuff through are going to control the rate at which we filter in the glomerulus. Tubulo-glomerular feedback mechanisms. Remember that macula densa and that group of chemoreceptors that were going to help regulate things based on the ion concentration in fluids? That's also going to affect glomerular filtration rate, how fast I suck things out of the glomerulus. So sodium chloride concentrations are going to affect the rate at which I filter things in the glomerulus. Some of the extrinsic, so that was intrinsic. What's intrinsic? Kind of internal. These are all kind of internal, but they might come from somewhere else in the extrinsic category. These controls that we just discussed are located right in the kidney, and they're part of what we call the autoregulation. They're right there already. Now, we have other controls that might not be right there in the kidney. What do we call those guys? Extrinsic controls. And extrinsic controls, we already know a lot of these already, are going to change what the cells do. The first one they talk about is sympathetic nervous system. And we know that the sympathetic nervous system kind of speeds things up sometimes, right? And we know that the nervous system can stimulate cells, like muscle cells, for example, to contract. And we know that myogenic controls are going to affect glomerular filtration rate. So if my sympathetic nervous system has an effect on my myogenic controls, am I going to have an effect on glomerular filtration rate? Yes? Everybody understand that? So my nervous system, based on different ion concentrations and hormone concentrations, chemistry of my body can also affect how quickly or how slowly I filter my blood supply. Then we have hormonal controls. Remember the Renin-Angiotensin Pathway? I told you we would talk about this again, didn't I? Well, here it is on steroids. So, what was the Renin-Angiotensin Pathway? What did that have to do with anything? Well, that's going to affect water regulation. It's going to control extrinsically what the cells do, and it's going to affect glomerular filtration rate. Yes? Now, I hope you get the gist of the fact that the rate at which I filter things might affect the volume of urine I produce. Yes? Now, why might I need to affect the volume of urine I produce? What's at the top of that diagram? We talked, Blood pressure. We talked about this when we talked about the circulatory system. And what was one of the things we kind of mentioned in passing might help control systemic, through the system, blood pressure? Yeah, and who's going to be helping me with that? The kidneys, the renal system. So here's some extrinsic controls that can control glomerular filtration rate. If my systemic blood pressure goes down, what can I do to bring it up? If it goes down, what can I do to bring it up? All of this crap in the middle. Yes? So this is how extrinsic controls can control the renal system to help control blood pressure in the system. So if I decrease my glomerular filtration rate, filtrate flow and sodium chloride in ascending loop of Henle decreases. The macula densa cells and the juxtaglomerular apparatus of the kidney are affected, because remember, those are the chemoreceptors that are going to give feedback. Vasodilation of afferent arterioles can happen. And I might increase glomerular filtration rate after that happens. Juxtaglomerular apparatus of the kidney. Remember, what am I talking about? Juxtaglomerular. What is that? That's a very hard word to say so many times. Remember, that's the nephron that hangs down more into the medullary region. That's the nephron that's going to help me with more what? Control of water balance. And these hormones, renin, angiotensin, aldosterone, are going to affect what those cells do with respect to water balance. Remember when we talked about these hormones? Okay. So they're going to affect in this pathway, and we kind of have to have one to have the other do what they're supposed to do. Renin, angiotensin, and angiotensin 2 are going to be released by the adrenal cortex. Aldosterone is going to come into play. It's going to affect the kidney tubules. So now I'm talking about those tubes where everything has to run. And what was running around all those tubes? Capillaries. Well, if I cause the cells in those tubes to increase the reabsorption, when I say reabsorption, what am I talking about? Stuff that I pulled out has to be brought back into the what? Circulatory system. So if I increase sodium reabsorption, read the words, water follows. So that's going to go back into the what? Back into the circulatory system. Remember those peritubular capillaries. That's going to help increase my blood volume. That's going to help do what? Increase my blood pressure. See how that all works? Now do we understand what the Renin-Angiotensin Pathway is? It's going to affect the cells and what they do during the filtration and reabsorption process in the kidneys. So if I need to increase my blood pressure, how can I do that really quick? If I need to increase pressure in the system, no talking over here. Yoo-hoo, back there. Yoo-hoo. If I need to increase the pressure in the system, what do I need to do, and how can I do it quick with respect to the kidneys? There we go. Put more water back into the circulatory system. More volume, more pressure. And why is pressure such a big deal? Yep, you need the blood to flow through the system, and then we've already talked about several different systems where we have to have some sort of pressure gradient for things to go across the what? Don't think cells, think tubes. We're inside the circulatory system for things to go across the what? Capillary beds, to go from the capillary beds into the tissues and vice versa. We have to have a certain pressure gradient in order for that to happen. Otherwise, things aren't going to move. So that's why it's so important to maintain the proper blood pressure. Know this diagram. Know the difference between intrinsic and extrinsic mechanisms that help control kidney function and glomerular filtration rate. Some of the other things that affect glomerular filtration rate. Prostaglandin. That's a vasodilatory paracrine, a hormone that affects cells beside. And what's a vasodilator going to do? Vasodilator, vasoconstriction. What's going to happen? When blood vessels dilate, things can move what? Faster in and out. When blood vessels constrict, I'm going to hold on to things more in the circulatory system. Then they talk about intrarenal angiotensin II, also another hormone that kind of reinforces the effects of angiotensin II. And what's angiotensin II going to do? Correct. Okay? And then we have something called adenosine. And what's that going to do? Yeah, it's actually it functions as a vasodilator. On the outline, look at what it says in the book. Although it functions as a vasodilator, systemically, adenosine constricts the renal vasculature. Okay? So in the renal system, and make sure you understand what that means. So in the notes it just says vasoconstrictor, but it's actually systemically a vasodilator. Okay? So don't get confused by that. Remember, we're talking renal system, so your notes are good. They're good. Just read your notes. So in the renal vasculature, in the renal system, which we're studying right now, it constricts. In the systemic vessels, it's actually a vasodilator. Why? I don't know. That's the way the cells react to it. The different cells react to it differently. So stick with those notes. That's why I pointed it out, because some of you actually read the book and you'll say, oh, wait a minute. So your notes are good. Not ignore it. Understand, understand what your note is saying as opposed to what the book is saying. Yes? Because it says it in the book too. It just says it differently. Does that make sense? Okay. Tubular reabsorption is what they talk about next. So, for sure, understand the renin-angiotensin and aldosterone pathway. They always forget about aldosterone. They just talk about renin and angiotensin, but don't forget that's involved in the pathway too. Understand that diagram. So, tubular reabsorption. So now, once I've sucked everything out, step 2 is to bring back into the circulatory system the things I shouldn't have sucked out. And that's what this diagram is talking about. So number 1, transport across the luminal membrane. What's a lumen? Yeah, so we're going now. Remember, we're talking about the tubes. We're in the nephron. That's the yellow thing on the left side of the diagram. Yes? And we're transporting back into where? The capillary beds, back into the circulatory system, some of the things that I should not have taken out. Now, one of the things that's going to help me do that is to reabsorb or to control how much the cells of the tubules reabsorb back into the circulatory system ions. And one of the major ones is going to be sodium. So a lot of these hormonal controls that help control blood pressure and water regulation are actually controlling the cell's ability to reabsorb back into the circulatory system ions like sodium. That's how they affect the pressure. Does that make sense? Hopefully. Somebody will nod. There was a nod over here. Any other nods? Good. Nodding yes. That's good. So, diffusion through the cytosol, transport across the basolateral membrane, and movement through the interstitial fluid. What's interstitial fluid? Yeah, fluid between cells. That's the bluish part. Back into the what? Capillary beds. So a lot of the controls not only control the cells of the tubule, the yellow stuff on that side, but they may also cause or result in vasoconstriction or vasodilation. If the blood vessels are constricted, they're not going to do what? Absorb things at as fast a rate back into the circulatory system. If these blood vessels are dilated, that is also going to affect how much of this stuff moves back into the circulatory system at any given time. So that's why we see in a lot of these complex diagrams, one of the effects of some of these is either vasoconstriction or vasodilation. That's also going to affect the reabsorption rate of some of these ions. And remember, who's going to follow things like sodium? Water. Water, nutrients, and ions. Going to happen through secondary active transport. What's active transport? Uses ATP to happen. So, it might not just be simple diffusion. We also have actual proteins inside the membranes of these cells that are physically going to take things from one place to another. That's active transport as well. So passive tubular reabsorption happens through little holes or pores called aquaporins. And what's an aquaporin? Aqua, water. And then we also have active transport that's going to help pull some of these ions physically. Protein receptors. In this diagram, we could actually see them. So on the bottom here, we see our aquaporins. And they're kind of like little water bridges that are going to help with water balance on either side. And then we see proteins that are physically going to grab things and put them back. Things that we shouldn't have taken out, like glucose. Do we want glucose in our urine? Hell no. We want it back in the circulatory system. We need that stuff, right? How about some smaller things like amino acids or some vitamins? Those got sucked out too. Do we want them to be sucked out? No. So during the reabsorption process, we have a combination of passive and active transport that are going to help bring those things back into the circulatory system. Again, things like glucose and amino acids, important vitamins, ions are going to get put back. And that's what this diagram is all about. So it's part of the reabsorptive process back into the circulatory system. And this says reabsorption in the PCT. What's the PCT? PCT. Ah, somebody knows. Say it loud. There you go. Proximal convoluted tubule. Where is that? It's in the nephron right after the glomerular capsule. So this is right after I suck everything out. I'm going to immediately start putting stuff back, reabsorption. A lot of reabsorption takes place in the proximal convoluted tubules of things that are important, like glucose, amino acids, and so forth. So understand this diagram. So we see two different kinds of arrows in this diagram, primary active transport, secondary active transport, and then we see a blue arrow, passive transport or diffusion. So through all of these different processes, reabsorption is going to take place. And much of that's going to take place right after we sucked everything out in the proximal convoluted tubules. I want to point out before I get too far, page 977, table 25.1. In this table, don't we love our tables because they make things small for us, and that should help us study. We see the different parts of the nephron and what happens in the different parts of the nephron. So different substances are reabsorbed at different times during their travels through the nephron.
So if you have high blood pressure and your doctor prescribes you a diuretic, that blocks the sodium reabsorption, so you get rid of water to bring down the blood pressure?
Correct. Yep. So that's how that's going to affect your cells, the cells of your kidneys. Yeah, correct. Correct. And certain substances can inhibit the release of antidiuretic hormone. Anybody know one? Caffeine. Alcohol. Once you break the seal, yes? And some of you know what I'm talking about. So some of those, that's how they affect renal function. So they're going to either stop or increase sodium reabsorption, depending on what needs to happen. Antidiuretic hormone is going to decrease or increase sodium reabsorption. Okay? So if I inhibit antidiuretic hormone, what's going to happen to sodium reabsorption? If I inhibit,
It's not going to be in as much.
correct. So what am I going to have more of?
You're going to get rid of more water.
Correct. More what? Urine. Yes? All right, so understand this diagram. That table will help you. Reabsorptive capabilities of the renal tubules and collecting ducts is basically what this table is. So it talks about the proximal convoluted tubules, the loop of Henle, which is just past the proximal convoluted tubules, where I'm going to reabsorb a lot of the water. We're going to do a little fine balance with my ions. And remember, sodium and potassium aren't the only ions. We have chlorine, we have calcium, we have magnesium, amongst others. And it's going to do that through osmosis, secondary active transport. Everybody see that in your book? And then we have the distal convoluted tubules. And what's going to happen in those guys? Again, a little fine-tuning. Fine-tuning with ions, sodium transport. We might even put some stuff back into the tubules that we sucked out. And then what's left is waste. And that's going to go into the collecting duct. And most of your urine is a waste product called urea. I shouldn't say that. Most of your urine is water. But most of the dissolved substances are waste, urea. But we're also going to get rid of some stuff that might be useful in our bodies, but we might have a little bit too much of. So some of those ions like sodium and potassium and bicarbonate ions or chlorine. And the other very important thing is, remember, those hydrogen ions. So if we have a little bit too many hydrogen ions in our blood plasma, that's going to mess with our what? pH. So what might I want to do with those extra hydrogen ions? Get rid of them in urine to help balance my body's blood pH. So that's another thing I'm going to see in urine as well. So that's pretty much the components of urine. Do I want glucose in my urine? No. I need that. That's important. What happens if I have glucose in my urine? Maybe I have too much glucose in my what? Blood plasma. Remember, I have to go through this little filtering process, and I only have so much time to do that. If I have way too much stuff to suck back in the proximal convoluted tubules, I might miss some of it. And it's going to end up coming out in my what? Yeah, I only have a certain amount that I can reabsorb in that given time. If there's too much in the blood plasma, you're going to end up seeing it in the urine. So a urinalysis can be a very informative tool to a clinician. Because if I start seeing those things in my urine, I know I might have too much of them where? In my blood plasma. And I won't have enough time to reabsorb them because I have too many of them. Does that make sense? Okay. So they talk about on page 978, the distal convoluted tubule and collecting ducts, ductus. And then step 3 is tubular secretion. And again, this is the fine tune. This is going to happen where? In the nephron. Where? The end of the line. Where? Distal convoluted tubules. So I dispose of some substances, certain drugs. One of the very important things that you have to take into consideration is renal clearance when you are developing a drug. So drug manufacturers have a lot of work to do, depending on what that drug affects. If your drug has a very low renal clearance, it takes way too long for that drug to leave the system, am I going to prescribe that drug very often? No. What if I have a drug that leaves the system very quickly? I'm going to have to prescribe that drug more what? Often. So they talk about disposing of substances in this section. We also refer to it as renal clearance. So for example, there's 2 pain relievers on the market. One's called Aleve, that's naproxen sodium, and one is called Advil. What's that? Anybody know what Advil is? Ibuprofen. They both kind of do the same thing, but you only have to take Aleve how often? Once a day, every 12 hours. Advil, on the other hand, ibuprofen, you have to take every what? 4 to 6 hours. Why? They're kind of doing the same thing, but why do you take one more often than the other? Renal clearance. Yes? You understand now? So that is an important factor when trying to determine dosage of different medications. Not only do you have to factor in how fast the drug itself will actually break down in the system, but you also have to factor in renal clearance time. So if it's a drug that gets cleared very quickly by the kidneys who are constantly filtering, I have to prescribe or use that drug more often. Or maybe if it gets cleared too quickly in the renal system, I'm not even going to bother. I'm going to move on and go try to find something else. Does that make sense? Eliminating undesirable substances. Urea, waste product. What else do I want to get rid of? Any toxic substances in my body. Get rid of excess potassium, controlling blood pH. That's the fine tuning that's going to happen in the end of the nephron line, and that's what tubular secretion is. I might have to bring some of this stuff back to get rid of it. So I sucked it out into the blood, that's reabsorption, but then I might have to put it back to get rid of it. And we fine tune in the end portion of the process called tubular secretion. So that's 3 major parts of the whole filtering process. So the nephron itself is actually making urine. The glomerulus is filtering that blood, and the nephron is making what? Urine. And the components of that urine are going to be dependent on what else is going on in the body. Yes? One of the things that's going to control urine volume along with hydrostatic pressure is something called osmolality. And osmolality is defined as the concentration of solute particles dissolved in water. Remember that whole discussion we had way, way back when about tonicity and the movement of water into and out of plasma membranes? Well, osmolality is kind of the same deal. Yes?
Well, I don't know, this diagram doesn't make sense to me because if it's isotonic at 300, which means it moves freely in and out of the cell, and then it gets higher as hypertonic, is it really hard to push the fluid?
No, it's because the nephrons, remember they're here, because of the filtration process. So the fluid as we go, that's what's going into the what? What am I finding in these pyramids? No, nephrons are here. Now is it making more sense? Nephrons here. So we did the whole filtering spiel here. Now as we move closer to the collecting area, minor calyx, major calyx, renal pelvis, what happens to the osmolality of the fluid? Do you think urine has more dissolved substances in it than? Yeah. So that's why the numbers go up. Yes? Because now all of those nephrons that are here are kind of pooling together in those collecting ducts, that highly concentrated, highly osmotic urine that's going to then leave. So that's what those numbers mean. Does that make sense to everybody else? Kinda? So think about what's happening. Remember, all those tiny little nephrons right in here are going to start pooling together. So all those little tubes, the collecting ducts, are going to start pooling together here in the pyramids. And all of that urine is going to start pooling together and going into this big tube. So that's why the numbers, the osmolality, is changing. Read it a few times. They talk about something called a countercurrent mechanism. And I'm not going to get too crazy about that. That's what this osmotic gradient in the renal medulla versus the renal cortex is talking about. Understand that this principle is principle of osmosis and osmolality, and it is going to help move substances through the renal system. So again, remember where the nephron is and what happens to osmotic balance as we go. Again, I'm not going to get crazy about numbers. There's a lot of numbers in these diagrams. I don't want you to get crazy about them, but I do want you to understand that this countercurrent mechanism is also going to play a role in setting up a gradient that's going to allow for flow into and out of the circulatory system and into the nephrons themselves. So countercurrent exchange and countercurrent multiplier is going to set up a pressure gradient difference with the help of osmotic pressure, osmolality, and hydrostatic pressure to move substances into and out of the circulatory system. And this area called the vasa recta around the nephron. So what kind of capillaries are these? No. They're around the tubes. They're peritubular capillaries. We see a shift at the vasa recta region of these peritubular capillaries, which will then shoot everybody into one of the major veins that leaves the kidneys. So, vasa recta is highly permeable to water. So we're going to have the ending water balance happening pretty much at that point, and to solutes. Nearly iso, what? Osmotic to interstitial fluid. What's interstitial fluid? Yeah, that's the fluid between these guys, right? There's a big space here, but they're actually right up against each other, right? So nearly isoosmotic to interstitial fluid due to sluggish blood flow, because we're kind of whipping up the corner. Remember, I go down better than I go up, so everything's a little bit slower in that vasa recta region. Blood becomes more concentrated as it descends deeper into the medulla and less concentrated as it approaches the cortex. So we're getting more water back into blood. This is kind of the end. And who's going to be responsible for that? This whole mechanism of the countercurrent exchanger and multiplier. And it's just basically movement based on differences in hydrostatic and osmotic pressure. Yes? Okay. So they get a little crazy with that. I don't know, she might have enjoyed the whole physics thing, but I don't. So that's what I want you to know about that. What's a diuretic? Yeah, it's going to change what those cells do. An osmotic diuretic, ready for this, is a substance that is not reabsorbed and that carries water out with it. So it's kind of like, it kind of acts like sodium. It attracts what? Water. So an osmotic diuretic is going to attract more water. What's going to happen to your urine volume? It's going to be greater. It's going to be greater because we have more what? Water. Yes? So, why might I be given an osmotic diuretic? Yeah, maybe I have too much pressure in the system. And again, let's go back to our previous discussion. If I want to quickly increase pressure in the system, what's the best way for me to do that? Suck more water back in. Remember, when I say system, I'm talking circulatory system. If I want to decrease pressure in the system, what's the easiest way for me to do that quick? Make sure more water stays in my urine. Okay, and that's what a diuretic does for me. Antidiuretic hormone does the opposite, so a diuretic sometimes does what? If I produce antidiuretic hormone to try and balance it out, what might a diuretic also do? Cause me to what? Decrease my production of antidiuretic hormone. That will also work to have me do what? Make more what? Urine, which is more concentrated with water. So we see absence of ADH, and this is the countercurrent multiplier and mechanism. This is how it affects it, but understand the principle. If I have no ADH, I'm going to have more what? A larger volume of dilute urine. If I have a lot of ADH, I'm going to have a smaller volume. And what's most of my urine going to be? Well, not most of it, but it's going to be lower in concentration in what? Now, the urea is still going to come out. Water. So a more concentrated urine is going to have more dissolved substances in it than a normal urine, which has a larger volume of water. When you wake up in the morning and you take your first void, is it pale? It's kind of concentrated, isn't it? Because what do you think you do during the night? You produce urine. You're constantly producing urine. It's a good thing you have a holding tank or you'd be peeing all the time, right? Yeah, but you produce less because your body, it knows you're going to sleep, and it doesn't want you to have to get up every 3 seconds to pee. Yeah, it holds on to more of the water at night, but you're still going to get rid of those wastes and things you need to get rid of. So in the morning, your urine is more concentrated because you produce a little bit more what? ADH while you sleep. Summary of tubular reabsorption on page 983. This is an awesome diagram because with pictures it shows you all the stuff we just talked about. So in the proximal convoluted tubules, 65% of filtrate volume is reabsorbed. So most of my reabsorption of the stuff that I sucked out in the glomerulus is going to happen in the proximal convoluted tubules. Okay? Remember, what's some of the stuff I'm putting back into the circulatory system that I shouldn't have sucked out? Glucose, amino acids, some vitamins. The descending limb of the loop of Henle, freely permeable to what? Water. This is where a lot of my water balance. Not permeable to who? Sodium chloride. So, am I going to do a lot of sodium chloride stuff in the loop of Henle? No, I'm going to do a lot of water stuff in the loop of Henle. Filtrate becomes increasingly concentrated as H2O leaves by osmosis. So what do you think I have to do on the way back up? The ascending loop of Henle. Hold on to the water. Do a little bit of ion balancing, sodium and chloride. Filtrate becomes increasingly dilute as salt is reabsorbed now. So this is where I'm kind of fine-tuning the water. And then in the distal convoluted tubules, sodium reabsorption regulated by aldosterone. So if I have more aldosterone, what am I going to reabsorb more of in the distal convoluted tubule? More sodium, which is going to then lead to the reabsorption of more water. I'm also going to mess with calcium reabsorption and chlorine and sodium, fine tune. What if I have low concentrations of aldosterone? What's going to happen in the distal convoluted tubule? I'm going to get rid of more water because I'm not going to reabsorb as much sodium. So this is where I fine tune that urine, and then what's left goes into the collecting duct. A little bit more water reabsorption's going on there, a little bit more, but just a little bit more. Just a little, because of the cell types. Renal clearance. We'll pick up with that on, what's today? Thursday. We'll pick up with that on Tuesday. Not on Monday.